Thank you for attending our webinar today. My name is Madhuri Ganta and I work in the customer service group at DNA 2.0. I'm filling in for Greg, who was not able to be here today. Please feel free to contact us at info@dna20.com with any questions or comments that you may have after the webinar. We will have a few minutes for question and answers after the webinar. DNA 2.0 is a gene synthesis company providing a suite of tools to help researchers go from DNA to whatever their end research goal may be. To facilitate this, we have a free software. It's called Gene Designer. I will demonstrate some of the features of the software today. We do research on the design of genes to determine how to improve expression of genes in various host systems. We do gene synthesis. We have several expression vectors that we can provide these genes in, and further down the line we do protein engineering and strain engineering as well. So why gene synthesis? Any gene you design can be synthesized. You're no longer limited by what's available in nature. Large amounts of sequence information are becoming available in GenBank every day. With gene synthesis, you can simply go to GenBank, pick up any sequence you want, modify it any way you want to, and send it to us for synthesis. So what is Gene Designer? It's a software tool that was developed by DNA 2.0, which allows you to design and manipulate sequences with ease. The software is free, has a graphically rich display with drag and drop features, making it easy to design genes, a customizable database of commonly used elements, and you can do sequence analysis, look for sequence motifs, restriction sites, ORFs, etc. In this webinar, I will show you how to download Gene Designer, go over some of the elements in Library Explorer, construct a sequence, and do things like search for restriction sites, motifs to demonstrate the capabilities of the software. I will show you how to use the cloning tool feature to perform in silico cloning, and then finally I will show you how to request a quote from DNA 2.0 for the gene, for the construct that we just created. So let's get started. You can download Gene Designer from our website. You go to Scientific Resources. Under Scientific Resources, click on the Gene Designer tab. Click Download Now. You will need to create an account. A registration key will be sent to that account. Troubleshooting and support are available. Scroll down to the bottom of the page, see a link to support, and you can check out our troubleshooting page here. So now that you've successfully downloaded Gene Designer, let's see how you can use it. We launch Gene Designer. On the left-hand side, you see a Library Explorer. On the right-hand side is your workspace. So in Library Explorer, we have new elements, a design toolbox, vectors, and a codon table library. New elements, we have new DNA element, new amino acid element, new ORF, new open reading frame, new construct, new folder, and new codon usage table. Design toolbox, this comes preloaded with several elements that are commonly used in gene design: prokaryotic regulatory elements, eukaryotic regulatory elements, fusion tags, vector components, etc. Let's look at some of the elements in these folders. So you see for prokaryotic regulatory elements, we have transcription, translation, and replication elements. So this, for example, you click on it, and you can see what this element is. So this is ribosome reinitiation spacer, and it tells you what you can use this for. It's to allow bacterial reinitiation in a polycistronic mRNA sequence. And the sequence can be placed between the stop codon of one protein and the RBS of the next. Vectors, we have some NEB vectors. This is the PT7 SNAP, and these are all the components annotated that are present in the vector. X-brain vectors, we have all the elements of 2 X-brain vectors listed here. And a codon table library with some standard tables. DNA 2.0's proprietary codon tables are not available with the free software, and you can import codon tables, and I will show you how to do an import of codon tables a little bit later. So let's start with the basics. Say you want to design a construct, an open reading frame flanked by 2 restriction sites. I have the sequences that I want to use in my construct on this text file. So you start, restriction sites, you use a DNA element, you drag it on to your workspace, give this a name. You can either enter the name manually or copy paste. So at the 5' end of my sequence, I am creating a Hindi 3 site. Next, I have an ORF. I'm going to call this a test ORF. And copy that sequence into the window here. You can see this sequence gets translated into an amino acid sequence. You can choose to keep the DNA sequence, or if you want to alter the sequence by back translation, you can remove this check mark here. For this construct, I'm going to keep the DNA sequence. You can add notes to this. I'm going to say this is a test. Click OK. Now the colors of these two are pretty close. I want to make these elements distinctly visible, so I'd like to change the color of this ORF. And you can do that simply by clicking on color and choosing any color that you want. Now at the 3' end, I'd like to create a SAC1 site. So I drag that on, 3'. So here I have a simple construct that I have created. You can import files to create a construct. For example, go to File, Import. I want to import a FASTA file from my desktop. I have the demo test sequences here, so I click that, click Open. FASTA import successful. So here's the same construct again. It's a 5' Hindi 3 site with an ORF. There's a test amino acid sequence in my FASTA file, which shows up here as well, and a 3' SAC site. So just a different way of importing sequence elements. You can import FASTA files or GenBank files. So let's continue working with the construct that was previously created. We are in icon view. There is a button here with which you can change the proportion of the sequence elements. So you can choose to view the elements in proportion to their size, or you can choose to look at them as equal sized elements. You can move elements easily. For example, I can move the Hindi 3 site all the way to the end of the sequence if I want to, but I actually do want it at the 5' end, so I'm going to move it back to the 5' end. Clicking on Sequence View allows you to view the sequence. This is a window that you can move to look at different parts of the sequence. You can change the size of this window if you want to scroll through larger sequence space quickly. And you just scroll through. And you see here we have a DNA sequence. This is an ORF. That is the DNA sequence that was used. And this is the translation. This is the amino acid sequence. And all the way to the end, we have a SAC1 site and an additional Hindi3 site. Now, I didn't want to actually create this additional Hindi3 site, so what I can do is click on it and delete it. I go back to icon view and make sure that I've actually deleted. Okay, so now I want to create an ORF that has a his tag. I want to convert this protein into a his tag protein. So what you can do to do that is choose, I want to insert the his tag after the start codon, so I choose the start codon and split. You can see now that the sequence has been split into two distinct elements. I want to insert an amino acid element in between these two. This is a 6 his. 1, 2, 3, 4, 5, 6. You can see a his tag has been inserted here. You can choose the codons that you want to use to make this tag. Now that I've inserted the his tag, I want to merge these elements back so I have one uninterrupted amino acid sequence. So what you can do is select sequences of the regions that you want to merge and say merge. Merge. Our software gives this element now this long name to show you the different sequence manipulations that we did. So if you'd like to, you can go back and modify this to whatever the final name you want it to have. Now that we have this construct, I want to make a note to myself that I inserted a his tag in this construct. So what you can do is choose the sequence corresponding to the his tag. And annotate it. So this is a general annotation. These are the bases that have been chosen. I want to say inserted his tag. So this information is available for me when I go back to look at the construct and see what manipulations I have made. If you want to create sequencing primers, there's different ways you can do it. Let's say this is the region in which I want to create a sequencing primer. You choose the region, go to Tools, click on Design Sequencing Oligos. I want the program to choose the best oligo for the region, and I choose double-strand oligo because I want a double-strand oligo. I can change the parameters of the sequencing oligo. I can change the size. I can specify a TN. I can specify a GC content, etc., and then say Create. You see now that the program has inserted sequencing oligos in that region. The program has found the best region to insert the sequencing oligo. Some of the other things that you can do, you see down here is a binoculars. This is a show find panel. Clicking on that opens up this window. You see manual motifs. my motifs, my restriction sites, open reading frames, repeats, and annotations. So let's look for restriction sites within the sequence. You click on my restriction site. Since I'm going to be cloning with Hindi 3 and SAC 1, I want to ensure that these sites don't exist within my sequence. So I say Hindi 3, I choose Hindi 3, SAC 1, and say find. So here's a list of Hindi 3 and SAC 1 sites that were found in the sequence, and you can click on it to actually see it in the sequence. So another interesting feature is this find potential sites. Say you want to introduce another Hindi 3 site into the sequence, somewhere in the middle of the sequence. What you can do is click find potential sites, click on Hindi 3 and say find. So what the program does is it finds sites where you can introduce a Hindi 3 site without changing the amino acid composition of the sequence. So just by changing codon usage, you can introduce these additional sites if necessary. So for example, at this site here, the program tells me that it's possible to introduce a Hindi 3 site. Hindi 3 is AAGCTT, so AAGCC. Okay. AAGCAT. Let's look at another site. We want to introduce this here. This is in the reverse direction, and I don't really want to introduce it there, so I'm going to go see if I can find another site, and you can see that it's possible to introduce this site here. This is AAGCA. So we've created an additional Hindi 3 at this site. You can search for motifs. You can either enter particular sequences that you want to look at and see that GCATT, for example, is present at position 11, and that position is highlighted. You can look for additional motifs. For example, if you want to look for additional Scheindel-Garneau sequences, click Scheindel-Garneau and say find. So you see no additional Scheindel-Garneau sequences are present within my construct. ORFs, you can look for open reading frames, and you can specify the kinds of open reading frames that you want to look for and the frames within which you want to find these. You can analyze your sequence for repeats, and you can specify the length of repeats that you want to find. So, for example, these are the repeats that are found in my sequence. And shown here is it's highlighted, the repeat. Okay. So we've done all this. We want to generate a report. So to generate a report, you go to Reports, click on Text Report, choose all the parameters that you want listed on your text report. You can choose whole construct, which is the sequence of the whole construct, elements, which is the sequence of each of the elements in your construct. You can choose a translation map, that's the translation of the DNA sequence into amino acids. You can choose to get a list of restriction sites, a list of motifs that are present in your construct, repeats. You can get the GC content of the whole construct of each of the individual elements. In addition, you can get the molecular weight of the ORF, the isoelectric point of the ORF. So you click Generate, and this is the report that has been generated. This is the sequence of the whole construct, sequence of each of the individual elements, a translation map, so this is the ORF translated into the amino acid sequence, a list of restriction sites, motifs, repeats, GC percentage of the whole construct and of each of the elements in the construct, the molecular weight of the protein, and the isoelectric point of the protein. So you can copy all of this onto a text document and save the report. To get a list of sequencing oligos, you go to Reports, say Text Report. I'm sorry. Go to Reports, Reports, Oligo List, and say Generate Report. Here we have a list of the sequencing oligos that we created. You can save this construct. So next time, if you want to reuse this or if you want to make modifications, you can open up that file again, and all this information will be available for you. You can add elements to this construct. There's several different ways you can do this. Click on this Add button, say New Design Construct, and say Test 2. You can add a construct simply by dragging a new design construct onto the workspace. Test 3. You can move elements within constructs. So say I want to make mutations in the first construct. So I first want to recreate the construct in each of my test constructs. So in this construct, if I want to mutate these two alanines into serines, I highlight the sequence, say split. You see this is now a separate element. I can click on this, and I want to make this two serines. Say OK, and choose the codons for this. And I want to annotate this so I know what I've done here. So I say serine. Can give it the position 245 alanine to serine. So you see the annotation right here. And now that I'm done with this, I want to merge this back. So I have one complete amino acid sequence. Again, you can generate a report for each of the sequences. Simply click on Report, just like I showed you, Text Report, choose all the elements that you want to generate the report for. Just some of the other features, let's go through the menus to see some of the things that you can do with this. We'll start with the Edit menu. Has the, you can undo. The last action that I performed was merge, so I can say undo. And you see that action, these elements are now separated again. So if I wanted to redo it, I just say merge. Cut, copy, paste, these are all just the general text editing features. you can find. It takes you back to the Show Find panel. You can group elements together. You can convert a DNA element into an amino acid element, or you can convert to DNA elements. This is converting an amino acid into a DNA element. You can clear codons from amino acid elements, fix codons, merge elements, delete elements. So let's clear codons from one of our constructs. So you click on this, say edit, clear codons. You see that all the codons in this construct have disappeared, so you can, for example, optimize the sequence and have the program pick different codons for you. Tools, back translate, cloning tool, design sequencing oligos, reports, configure. You can create a group of restriction sites that you want to use in the Find panel. So you click on restriction sites. So shown here is the default group. You want to create your own group. Let's call this new group. You can, these are all restriction sites, and these are the restriction sites that are currently in this new group. So if you wanted to modify it, if you wanted to add, you simply drag and drop the sequences, the restriction sites that you want to include as part of your group. Now say one of these is something that you dragged on by mistake and you don't want to have this in your group. You simply drag it to trash. Okay. And have that sequence disappear. So you created your own group, and in this Find panel, when you go to restriction sites, you can choose your group, and it lists the sequences that you've chosen. Okay, so I'd like to show you the cloning tool now, and then briefly show you how to do a back translation as well. So let's start with the cloning tool. I have a file that I've created previously that I'd like to use, so I'm going to close this. I don't want to save changes. I'm launching the file that I had saved previously. So I go to Tools, Cloning Tool. I want to clone something into our Ramex vector, so I choose the vector. I want to use SAL1 and XBA for cloning. So I click on SAL1, click on XBA1, choose and use cut. So you want to cut the fragment with these two restriction sites. This is the fragment that results from the digest. You drag this on to your final clone. This is the construct that is going to be my insert, so I drag it into the cloning tool, and I want to use NDE, BAM1, and ZO to digest this. So choose. So I see NDE, ZO, and BAMH1, and these are the positions that they're present at. So I choose the enzymes that I want to cut with. This is the fragment resulting from the digest. Let's see if we can clone this into our Ramex vector. So I drag this on to the final clone and say clone. So you can see here there's a message that the segments had to be flipped to accommodate the cloning, and that's okay for me for this project, so I'm going to say okay. If you want to add another piece, you can say use cut. So I've chosen now NDE and ZO. This is the fragment resulting from the cloning. I drag it on here and I say clone. Since the ends do not match, the program tells me that the ends do not match. So this is a very useful tool, especially if you're trying to put together several fragments to sort of test your design in silico and make sure that you've got ends that match and cloned in the right direction. I don't want to use this fragment, so I'm going to drag it to the trash. And these are the two fragments that I want to create the clone with. So I say clone. This is the final construct that's created. So I drag this on into Gene Designer, drag this back to Gene Designer, and I can look at the sequence and make sure that this is the end result. This is the sequence that I want. So very useful in silico cloning tool. Now that I've created this construct, you want to submit this. If you want to submit this for a quote request, you go to Tools and you say Order, Gene Request. This is the name. You can choose the vector that you want this applied in. You can tell us if you want a large-scale plasmid prep and if that should be endo-free. Rush delivery, you can add notes. Log in to your account. With the password and say request quote. This is the status of your request. The request has now been submitted. You see a reference number is generated. This is your reference ID. If you want to call in with questions about this quote, you can use this reference ID. So this is your confirmation that we have received the quote request. Okay, so you can do back translations with the software. You can do optimizations with the software, but the codon tables that DNA 2.0 uses in-house are proprietary and not available with the free software. So to get the best optimizations, it's best to send us your sequences. We'll do the optimization for you, and there's no additional charge for optimization. So having said that, let me show you how you can use this software to do optimizations. So this is my sequence. This is an amino acid sequence, and the codons have been chosen. I want to demonstrate how to use the optimization, so I'm going to clear the codons from this element and have the program pick for me the codons that are best for expression in my host system. So you can go to codon table library. You can import any codon table that you wish to have, so click on import it. I already have here imported a bacillus table, but I want to use Pseudomonas for this construct, so I can import a new codon table library. So I go to File, Import, codon table. You type in the first 3 characters of your host system. You can see a long list of Pseudomonas genomic codon tables that are available. This is the codon table that I want to use, so I click on that and say import. So here now is the Pseudomonas codon table. So to use this table for optimization, I first need to make a back translation profile. So I go to Configure, Back Translation Profile. I'm going to modify this because I want to make this a Pseudomonas table. So give it a different name. Drag the codon table that you want to use into the codon usage table. The threshold is a threshold for rare codons. You can add motifs and restriction sites that you want to avoid when you do the optimization. So for example, I want to avoid APA1. I want to avoid creating BCL1 sites, maybe BAMH1. So you choose the list of restriction sites that you want to avoid during the optimization. Now say I've realized that I've dragged APA-L1 by mistake, so I can simply move this to trash to remove that sequence. You can edit the length of the repeats to remove. You can set that to 10 and close. So I've now created a back translation profile. To actually use the back translation profile, go to Tools, Back Translate, choose the program that you want to use, and say Back Translate. You see here, the program goes through several iterations trying to match codon usage in your sequence to the codon table that was selected. So let's give the program a couple of minutes to finish this optimization. Since this is a long sequence, I'm going to say accept current best and stop at this point. So accept current best. So the optimization is done. You can see that codons have been chosen, and you can generate a report. Again, you will need to choose all the parameters that you want listed in the report. And here is a report. So very useful software, very powerful and easy to use. I want to thank you for attending. Please feel free to contact us at info@dna2o.com if you have further questions. I just want to also alert you to several upcoming webinars. You can find more information about these on our website. You go to company news events, news and events, and you can see a list of upcoming webinars. So thank you again for your attention. info@dna2o.com. Please make a note of this email if you want to send us some questions. Thank you.